immunoglobulin fold in constant domain of immunoglobulin G has two layers of beta, one 3 beta and the other with 4 beta. The sheets are closely packed against each other, and even though the actual structure consists of two separate beta sheets, it is convenient to regard the topology as a form of Greek key barrel. So you can have the name of each beta: A, B, C, D, E, F, G. Loops are short. And structure is the same in both heavy and light chains. The loops may vary in length and sequence among immunoglobulin chains of different classes, but are constant within each class. The sequence of the loop is invariant. The function of this loop are not known, but they are probably involved in the effector functions of antibodies. When an antibody-antigen complex has been formed, signals are mediated through Fc regions to different systems, such as phagocytic cells or the complement system, which will destroy the complex. These activities are triggered by specific interactions of ligand with the constant domains of immunoglobulin molecule. The overall structure of the variable domain is very similar to that of the constant domain, but there are 9 beta strands instead of 7. Here you can see the comparison of the structures of constant and variable domains of immunoglobulin. Beta strand labeled A through G have the same topology and similar structure. There are 2 extra beta strands, 2 C' and C2' in red color in the variable domain. The loop between these 2 strands contains hypervariable region CDR2. The remaining CDR regions are at the same end of the barrel in the loop containing beta strand B and C CDR1 and G and F for CDR3. A disulfide bond bridges strand B in one sheet with the strand F in the other sheet in both the constant and variable domains. In B at the bottom, you can see the beta strand viewed end-on, illustrating that one beta sheet has the same 4 strands in the 2 domains, blue one. The specificity of immunoglobulins is determined by the sequence and size of the hypervariable regions in the variable domains. As you can see this structure, the hypervariable regions are clustered in loop regions at the end of the variable domain, the top position. Examining the loop contributing to the 3 CDRs, the loops from 5-stranded beta sheet contribute more residues to the antigen binding sites than the other 4-stranded sheets. All loops in a 5-strand site contribute. Look at the red one. While only one from the other. Another interesting thing, Regarding this structure, the fact that there exists a great number of sequence variation in the loop regions of antibodies demonstrate that the folding of the structural framework is quite insensitive to changes in the functional loop regions. Thus, it has been possible to construct, using recombinant DNA method, chimeric antibodies carrying CDR loop regions from one antibody and framework regions from another antibody. The variable and constant domains in the light chain of immunoglobulins are folded into two separate globular units. The CDR regions are at one end of this elongated molecule, here, red one, in the FAB framework, as well as in the intact immunoglobulin molecule. Domains associate pairwise so that VH interact with VL, CH1 with CL. By this interaction, the CDR regions of both variable domains are brought close to each other and together form the antigen binding site. Constant domains associate by interactions between the four-stranded beta sheet in both CL and CH1, which are closely packed with mainly hydrophobic residues in the interface. The variable domains associate in a strikingly different manner. In the variable domains, the five-stranded beta sheet form the domain-domain interacting area, as you see in B. Furthermore, the relative orientation of beta strand in the two domains is closer to parallel than in the constant domains. Only 4 of the beta strands are involved in packing the variable domains against each other. The strand C2' is not involved. The diagram in C illustrating a hypothetical packing of the variable domains through their four-stranded sheet. The hypervariable regions would be far apart in comparison to real structure in B, which shows a closely localized 6 CDRs, red one. The curvature of the 5-stranded beta sheet is such that they do not pack tightly against each other. Instead, each sheet forms half a barrel, where the 6 CDR loops are positioned at the same end of the barrel. This one. Hydrophobic side chains fill the inside this barrel. Drawing of a specific filling model of the hypervariable regions of an FAB fragment. The superpositions of 5 sections are shown, cut through a model as shown in B. It is clear, seeing that all 6 hypervariable regions contribute to the surface shown here. This can create flat, extended binding surfaces for protein antigens or specific deep binding cavities for small hapten molecules. The specific binding of hapten, in this case phosphorylcholine, to the FAB fragment has been studied by David Davis group at NIH. The binding cavity is in a cleft between the light chain and heavy chains. A number of side chains from the CDRs except CDR2 bind phosphorylcholine in a manner that is very similar to that of a substrate or inhibitor binding to an enzyme. The interactions of antibodies with protein antigens were studied by Roberto Bourjac in France in 1986 by using lysozyme. The shape of the interaction area between lysozyme and the CDR loops of antibody is easily distinguished from the hapten binding cleavage. The interaction extends over a large area, as shown here. FAB fragment, yellow and blue, and the antigen, lysozyme, in green color, have been separated in this diagram, and their combining surfaces are viewed end-on. Atoms that are in contact in the complex are colored red both in FAB and lysozyme, except glutamine 121 in lysozyme, which is violet. These residues packed against each other with a density similar to that found in the interior of protein molecules. For example, glutamine 121 in lysozyme protrudes from the lysozyme surface and fits snugly into a crevice between the CDR3 loops of VH and VL. Interacting area is contributed largely by CDR3, which is very significant because CDR3 of heavy chain is a site for the somatic mechanism for generating antibody diversity. The structural changes observed for antigen and antibody in separate form or as a complex, so the binding could be explained by log and key model in this case. However, in other cases, conformational changes were observed, so the induced model also applicable in antigen and antibody binding. To engineer antibody, this kind of question is going to be valuable. Can the structure of CDR loops vary randomly, or are there certain preferred conformations that can be deduced from the lengths and sequences of these regions? It turns out 5 CDRs except the heavy CDR3 have only limited repertoire of main chain conformations. For example, L2, which is a 3 residue long, virtually have a single conformation. H2 might have one of 4 conformations called rotamer. In contrast, L3 is very difficult to predict and model correctly because it varied in length and sequence.